Hi guys, back for another lecture, this time picking up after digestion on pretty much what we do with those nutrients that we acquire through the digestive tract. And so this topic will be general metabolism. And so what I'll do is I'll do this video on general metabolic pathways just to review, make sure everybody's up to speed with what the basics are. And then I'll come back with another video that talks about the hormonal regulation of these same pathways. So I'll plug that in after we've got the basics down. And just as foreshadowing, the key hormones for controlling metabolism are insulin, and again, metabolic function in general, nutrient use, nutrient storage, that sort of thing, would be insulin, glucagon, cortisol, growth hormone, and epinephrine. I think I got them all. Yep. They're at least the primaries. Okay, so let's go ahead and start with sort of like a basic picture, a review of what the nutrient pool looks like, and then we'll get into an explanation of largely of catabolic pathways, but along the way we'll point out anabolic ones as well. So I want to we'll start with something really generic. So if we start with a cell, right, nucleus, a cell has available what we call its nutrient pool. And these are the nutrients from which it can partake in order to either make ATP or build larger macromolecules or polymers of some sort. And so this nutrient pool can be broken down, so kind of like two things. I'm going to shift it up a little bit, actually. And we can take it, we can kind of do two things, right? We can be catabolic with it, or we can be anabolic with it. Right? Those are the two general divisions of metabolism: breaking things down, releasing bond energy, and building bonds and larger molecules, requiring energy input. And so when we're catabolic, well, in general, when we undergo these reactions, we release heat, right? Energy exchanges are never very efficient, and so heat is always released. We're always increasing the entropy of the universe. But we're going to harness some of that nutrient pool bond energy when we're catabolic in the form of ATP. And that ATP can be utilized, and again, it's heat, it's going to dissipate off into the universe. That ATP can be utilized to drive the negative free energy change, the positive free energy change when we undergo anabolic reactions. And again, that bond energy exchange is never efficient, and so we have more heat release. And now we're creating more order, at least in this cell, right, in that particular system, by creating lower entropy, highly structured and organized macromolecules and membranes and things of that sort, right? And so we can basically go one of those two ways. And again, like I said, we'll focus on this one for the moment, and then we'll point out some of the places, some of the key anabolic pathways and mechanisms, probably which you're maybe even more familiar with, like protein synthesis from introbio and cell bio and other courses you may have taken. Okay, so let's take this nutrient pool. Well, first of all, we're going to back up. We're going to say this is the nutrient pool of a cell, but there's a nutrient pool that the whole body has. And if you had me for A and P, you may have heard this before. But again, it's a good introduction for everybody. Make sure we're all on the same starting point. So whole body. We call these nutrient stores. Right? So these are the nutrients we have available for energy, if you will, and again, or interconverting them and then being anabolic, right? We can be catabolic in order to be anabolic, break something down to build something else. That happens frequently. Our primary energy source, again, for not all, but for many cells, is carbohydrates. Most cells use multiple energy sources, but a primary tends to be carbohydrates. And that actually turns out to be our smallest nutrient pool. And so, again, it's variable fairly dramatically between people, but even if it was 100% difference between the highest and the lowest, it's not going to come close to some of these other nutrient pools. And so I'm going to call it about 1,500 kilocalories. A kilocalorie is what we call a calorie. And again, that's approximately what a person's daily caloric consumption is. Again, with significant variation around that. So we've got about a day's worth of carbohydrates stored at any given moment. Let's go to protein, just because it's the second largest store. And again, highly variable differences between male and female, and certainly dependent upon body size, that sort of thing. I'm going to put it at about 35,000 calories. And again, you could say 50 and you wouldn't be out of the realm of normal. So significantly more. But remember, DNA only codes for one molecule type, and that's protein. So these molecules are intentionally constructed and evolved to serve a particular purpose, whether that be a structural purpose or a functional purpose like enzymes and channels and things like that, transcription factors. And so this is not an energy pool that you really want to dip into in terms of our proteins. Now, if you eat excess protein in your diet, we will certainly utilize those amino acid, the energy of those amino acids. Remember, we absorb as monomers, collective digestion. We would use the energy of those excess amino acids catabolically to make ATP. Now, we could also modify them and then become anabolic and make extra fat by eating too much protein or any type of nutrient. Any excess will can find its way into fat storage. Which brings us to fat, or lipids. And I'm going to put about 150,000 on that, right? So dramatically higher than the carbs. You have to think of the lipids, not that we don't use them on a regular basis, but they are not the primary, quote-unquote, gas of many cells. There's an exception to that, at least at rest, which is skeletal muscle. Skeletal muscle does tend to use lipids at rest for energy, more so than carbohydrates. But you have to think of the lipids as an evolutionary solution to fasting, right? It is not intended to be the primary energy source for most cells. We have this huge caloric store so that if we don't have food for 2 weeks, we don't starve to death. That doesn't happen very often in civilized society these days, but evolutionarily it makes a lot of sense. We may think that we have excess body fat, excess lipids. Many of us do. You might think, oh, gee, I'd like to get rid of this excess, these excess lipid stores. Keep this in mind. They are, right? The efficiency here is dramatically better than in terms of carbohydrates. So if you had to store this many calories as carbohydrates, you would weigh much, much more than you do right now. Because A, this is not hydrated, and these are. And B, we more efficiently store calories in this form than in this form. If you look at the grams per calorie on the back of your cereal box tomorrow, you'll see that the fats are more than twice the calories per gram than carbohydrates or proteins. So very efficient energy store. but very large nonetheless. Okay. I want to back up just a second, maybe not back up, but also talk about other energy sources that cells have. And those would be the intracellular sources at any given moment. And some of these can be intracellular. Well, all of these tend to be intracellular to some degree. Some of them get shuffled around between certain storage sites, like lipids tend to be stored at the liver and fat tissue. Carbohydrates stored at the liver, right, and we get dispersed out to other tissues in times of need. But an energy source that all cells have at any given moment is ATP. And I don't want to put that in terms of calories. We could. I would rather put it in terms of like a duration of how long it would last if we did not replenish the intracellular ATP. And that's somewhere in the ballpark of 2 seconds. Right? Remember I said this is about a day. Your cells, individual cells, have about 2 seconds worth of ATP. So we better be making more, because as soon as you stop making ATP, muscle cells stop contracting, the sodium-potassium pump stops, protein synthesis stops, everything stops, right? So we never run out of ATP, right? We never run out of ATP, or at least not for any remotely significant period of time. The backup to ATP is creatine phosphate. Creatine phosphate is also contains high energy phosphate bonds, right? That's where the energy is stored here, is in the terminal and even secondary high energy phosphate bond. But we get most of it out of that first one, going from ATP to ADP. And creatine phosphate, we also have a high energy phosphate bond, and we can transfer the energy from that bond to ADP. Sorry, mute my phone. And how much of this do we have? Again, so in terms of time, I would put it in the sort of 15 seconds realm, 10 to 15 second realm. And so our cells, even if we don't make new ATP from our nutrient pool, right, our whole body nutrient pool, our cells could last, 15 to 20 seconds without it. And so what I like to say sometimes is that, the world record in the 100-meter dash, close to the 200-meter dash, but at least the 100-meter dash, they could be breathing pure nitrogen, not making any ATP aerobically from these sources, relying essentially only on ATP here. You could get anaerobic ATP, but even if you let that out, right, fermentation, leave that out, I don't think the world record in the 100-meter dash would really change significantly. You just don't need metabolism, right, during that short time frame, because most of what the cell is going to burn through is already available. Right? Okay, and again, yes, it's going to be using it very quickly, but this is 2 seconds utilizing it sort of as a sole source of energy. Okay, so let's start by examining catabolic pathways, and again, we're going to start with carbohydrates, because carbohydrates are a good example, because A, they are a primary energy source for many cells, and B, many of the other nutrients funnel into the carbohydrate metabolizing pathways, so that it becomes a little bit redundant. All right. So we're going to start with the storage polymer for glucose, right? Glucose being a carbohydrate, and that is glycogen. Not very many cells store significant amounts of glycogen. It's largely found in liver and skeletal muscle. Skeletal muscle, because it utilizes so much energy so quickly that they have sort of an on-site storage, very significant storage of glycogen. The liver tends to be sort of back up to that. As during more prolonged exercise, the liver will start to release some glucose as well from stored glycogen to sort of back up the skeletal muscle stores. But the skeletal muscles actually store more glycogen than the liver does. It can be degraded or built from its monomer, glucose. And now we should put names on these, and this is going to be a tight figure, so I have to kind of really erasing things and sort of adding new wings to the pathway here. When we're building things, the pathways tend to have an end in genesis, right, making. When we're talking about the catabolic pathways, right, anabolic would be upwards here. Catabolic tends to end in lysis, which means to break, right? Those are going to be the downward arrows here. And so this would be glycogenesis. And this arrow would be glycogenolysis. Right? Just to name the path, because again, when we start talking, bring the hormones in and which pathways these hormones stimulate or inhibit, we need to know the names of the pathways and understand what those terms mean. So now we're down to the monomer glucose molecule. Glucose is readily dispersed and carried by the bloodstream, right? Taken up, its uptake is driven by insulin at skeletal muscle and adipose tissue. It's taken up by other cell types in a non-insulin dependent fashion. As I said before, many cells will use glucose or carbohydrates specifically, carbohydrates, glucose specifically, as their primary energy source. And so we can break down that glucose, and we're being highly simplifying, like lots of enzymatic reactions here, into pyruvate. Or pyruvic acid, if you prefer the dissociated versus the non-dissociated acid version. Glucose is a 6-carbon molecule, right? It's a hexose. Pyruvate is a 3-carbon molecule, so you get 2 of them. Right? 2, 3 carbon molecules, I guess is the way to write it. And let's see, so this pathway is obviously glycolysis. Right? And we'll get to the sort of the metabolic, I've kind of written out the metabolic gist of that one already, at least one of them. This one, making glucose, is called gluconeogenesis. And it's called gluconeogenesis, neo for new, and genesis for making, so making new glucose. And typically what we're making it from is either a non-carbohydrate precursor, so we'll funnel in amino acids and some other molecules here later on, or we could be making it from lactate, which again, we'll show you in a minute. But again, so making new glucose from some non-carbohydrate precursor. And I know lactate can come from a carbohydrate in the first place anyway, but usually they state it as coming from a non-carbohydrate precursor. And that's gluconeogenesis. Okay. This pathway tends to take place, again, sort of predominantly at what I call metabolic hubs, like the liver is a primary site of making glucose. The kidneys actually are a site of making glucose. They do a significant amount of gluconeogenesis. Many cell types don't have the enzymes capable of running gluconeogenesis. Remember I said the liver is like a metabolic hub where it'll take nutrients sort of released from other locations and interconvert them, store them possibly, but maybe interconvert them into other things, lactate into glucose, it's called the Cori cycle, and then put that glucose back out in the bloodstream to be utilized. All right. So now let's say, we have to say a little bit more here about glycolysis, because glycolysis has its key output when we are aerobic, in other words, in the presence of oxygen, is pyruvate. And the rationale for that, which we'll get to in a minute, is that it's the substrate for the next series of reactions. In the absence of oxygen, then the pyruvate is going to get shuttled. I should say in the absence of oxygen, or when ATP demand exceeds the ability of the aerobic pathways to supply it. Focus on that for a second. Glycolysis has greater quote-unquote power output, energy output in terms of ATP production than does aerobic metabolism. Yes, it is much less efficient. You've heard that before in intro bio, right? You only get 5% of the energy anaerobically out of glucose than you do when it's aerobically metabolized. Do not think that that means you get ATP out of aerobic metabolism faster. You do not. Again, this is sort of like a misconception that sort of permeates a lot of the intro metabolism. What that means is that when we go from pyruvate to lactate, and we can go backwards, as I said before, right, from here to here to here, that would be running gluconeogenesis. But when we're predominantly going to lactate, we're doing so because the ATP demand is exceeding what the aerobic pathways, which we haven't seen yet, what the aerobic pathways can provide. This is the higher ATP output, more ATP produced per second through glycolysis than through oxidative phosphorylation, again, coming up in the mitochondria. So what we need to add here, though, is a sort of key output is ATP. It is only net 2 ATP. It is not much, right? You get a theoretical net of 36 when you do it aerobically, right? So 5%-ish of the ATP that you would get aerobically. But you can produce it faster. You can produce more ATP more quickly this way, anaerobically through glycolysis, than you can aerobically. And so again, when ATP is limiting, you're going to end up going this way. Now, you could also become oxygen limited, and that will minimize our ability to go in the other direction as well. That's a really rare scenario. Okay, why do we go from pyruvate to lactate? It's really a redox balance thing. If you look at one of the components of one of the outputs of glycolysis here, it's NADH. In order to continue to run glycolysis, we have to get back to NAD+. That's what this conversion does for us. And so that NAD+ goes back, becomes NADH again, gets converted to NAD+ and back. And so what going to lactate allows is glycolysis to continue. It's the only point of doing it. You're not getting any more ATP by going from pyruvate to lactate. It's about redox balance and allowing glycolysis to continue. Okay, now, could you potentially have an acid-base problem because of lactate production? Yes, it's actually not. And again, there's another misconception is that fatigue and metabolic acidosis comes largely from lactic acid production. Lactate would be the anion, right? You would also get H+, put a minus sign on that if you want. Again, not a misconception. That's not where most of the acid is coming from. We won't get into that right at the moment. Right? Kind of a side channel on this whole process. Right? So let's put. I can put no O2 here. Yes, that's true. Need ATP. Sorry, that's small. Fast. Right? We need ATP fast. If ATP, maybe a better way to put it would have been ATP demand is high. We're going to end up going this direction as opposed to this direction. And again, that would be O2, low O2 demand. I'm not going to say low. That's lower O2 demand. Right? Only when O2 demand is super high do we end up going this way more dramatically. Or if O2 is lacking. Right? So if O2 demand is lower, then we can supply ATP fast enough through the aerobic channels. Right? Where's this arrow going? It's going to the mitochondria, which I don't have room to draw. So I have to erase and start over again. Okay? So that's where I'm going next. We're going to follow our pyruvate, our 3-carbon molecule, into the mitochondria. All right, here we go. And again, right now we're only looking at the carbohydrates. And glycolysis can only process carbohydrates. But we're about to show you the Krebs cycle and oxidative phosphorylation can metabolize the other nutrient types as well. So remember that they're going to funnel in to some of these places. And when we start to go anabolically, then they can funnel in and go back, right? We can use amino acids to make glucose and to make glycogen. So they can actually funnel into anabolically. They just can't funnel into it catabolically. Only carbohydrates can be funneled through glycolysis catabolically downward. All right, so let's draw you. I think we hit some of this before, but because we talked about, I remember we talked about thermogenin, I think, at one point, and I sort of remember drawing the basics of this. So here's a pyruvate. Oops, spelled that wrong. Pyruvate. Right? This is the cytoplasm. That's where the glycolytic enzymes are found. And pyruvates are mitochondria. Is this double-walled, right? So this is the mitochondria. This is double-walled, double-membraned organelle. And a pyruvate is taken up in a fairly fancy fashion with some carriers and channels. And along the way, it's being processed, and it's actually going to have a carbon dioxide removed from it upon entrance to the mitochondria. And so it goes from a 3-carbon, right? And this is going to happen twice because there's 2 of them. It's going to go from a 3-carbon to a 2-carbon. Well, a 2-carbon group is an acetyl group. And it's going to get associated with, become stuck to a coenzyme A. And so we end up with acetyl-CoA, right? And again, that's a 2-carbon molecule, right? So 3 carbons, here's one of them. We still have 2 of them over here. Again, this whole thing's going to happen twice. We want to follow those just to sort of like, for the stoichiometry, keep track of where all of our atoms are going. And the acetyl-CoA is what enters the Krebs cycle, or the carboxylic acid cycle, or the whatever else, the citric acid cycle, right? Same thing. There it is. No enzymatic reactions required. Well, they're required. You're not required to know them. You're required to know what the metabolic gist is. What's the key output, right? For glycolysis, it was lactate. Well, it's ATP with the associated production of lactate when your ATP demand is high and you're anaerobic, and it's pyruvate when you're aerobic and your ATP demand is lower, right? And so we're on our aerobic pathway here. So you added a 2-carbon group here, so you have to get the 2 carbons out of there because they're going to come back and start all over again. And so the end intermediate in the Krebs cycle is oxaloacetate, which is a 4-carbon molecule. And so you added a 2-carbon, it makes it a 6. And so we have to get back to a 4. And so along the way, right, we're going to give off 2 more CO2. Go ahead and exhale that, right? Make sure that our acid-base balance stays normal, right? Because when CO2 builds up, it combines with water, becomes hydrated, carbonic acid, dissociates, makes you acidic, right? So you have to get rid of this in order to maintain acid-base balance. Okay, that's important, but doesn't have anything to do with that. It's a waste product in this scenario. We do get a little bit of ATP out of here. It's actually GTP, but it's equivalent high-energy phosphate bonds, so we'll just call it ATP. Again, minimal, but similar in numbers to the amount that glycolysis was making for us. The key outputs here, and I'm just drawing them over here randomly, are NADH and FADH2. And what it is what we're doing is we're sticking to hydrogen atoms. NAD and FAD are electron transport molecules, and really it's more than the electrons. It's carrying the protons too. It's just that it's not necessarily one-to-one. Whatever. I like to call it a hydrogen atom carrier because a hydrogen atom is a proton and an electron. It's just not always one proton and one electron. But okay, details. Our hydrogens are coming out of these organic molecules. There are the oxygens. Sorry, there are the oxygens. There's the carbon. Where's the hydrogen? There it is. Right? So what are we going to do with the hydrogens? Well, what we're going to do is we're going to take the electrons and we're going to pass them molecule to molecule in a series of redox reactions, which is going to result in a negative delta G energy release, exergonic, and energy release, which we're going to use to transport the other half of the hydrogen atom, the proton, into the intramembranous space. So these guys, so again, the metabolic gist of the Krebs cycle, the catabolic gist of the Krebs cycle is the hydrogen atom removal through the production of these two. All right? I'm going to take these guys. They're going to come up here. We'll add a little color. We have our cytochromes. Right? And we're going to take the electron. We're going to pass it and pass it. And some of those reactions where the electron gets passed, the delta G change is large enough that we can push a proton across the wall here, across the membrane. And so we're going to end up with pluses, right? Remember the plus indicates a free proton. So it becomes a very acidic space. Let's have our electron pass all the way to the end here. Right, so there's the electron, there's the proton. What do we have? Well, we have stored energy. We don't really have energy that you can hand out to the sodium-potassium pump yet. Right? We have energy like, potential energy in a gradient sitting here. It's why it's called chemiosmotic phosphorylation. It's a chemical gradient storage of energy, and it's osmotic because we're putting a solute on one side of the membrane. We're going to allow it to come back, though. We're going to allow it to come back across a channel that's an enzyme that phosphorylates ADP, right? So that's ATP synthase or synthetase. Right? So these guys are going to come back across. Just going to show you two of them here. Right? And in the process, in red, we're going to make lots of ATP, right? You're going to make 90% of the ATP of this whole aerobic pathway, right? A little bit here, a little bit over in glycolysis, lots right there, right? And again, you need the precursors. You need ADP, and you need inorganic phosphate. And the energy, the kinetic energy, due to dispersal or dissipation of that potential energy gradient is harnessed to phosphorylate ADP. Okay, so there's your key output of the oxidative phosphorylation. Well, just to finish the picture here, electrons are passed at the end there. You can't just pile up electrons on one of the cytochromes. It's got to give them to somebody. It gives them to molecular oxygen, so you end up with oxygen-free radical. Right? So again, why you breathe in, why you breathe out. Right? Here's your oxygen-free radical, the O dot. Right? Well, that's highly reactive. The good news is we have something for it to react with, which is the two protons. Right? And so you end up with H2O, which is metabolic water. Right? So you make water from the food you eat, right? From the hydrogen is essentially from the food you eat, and the air, the oxygen from the air you breathe, we make metabolic water. And it's not a lot, it's a couple hundred milliliters a day, but it's water that you did not drink and it did not come in with the food you ate. You literally produced it. Okay, so we got the metabolic gist of our Krebs cycle, right? Output of these two guys, carrying hydrogen atoms. The most important component, I guess, is the electron, since that's what everybody calls it. And I get why they're not calling it hydrogen atom carrier, because again, it's not, hydrogen atom is one proton and one electron, and it's not always, they're not always being carried one to one. But you get the idea here, where we're taking the electron of that original hydrogen atom and the proton, and sort of like doing two different things with them. One's used for energy sort of dispersal, and the other's sort of like acquiring that energy through the formation of a gradient, the proton gradient. Okay. Now what we need to do is funnel in our other nutrients, right? Because so far we've only really talked about carbohydrates, and I need to talk about how fats or proteins are metabolized so that when we start talking about the hormones, we can bring in, right, fat-sparing or glucose-sparing hormones and know what we're talking about. I'll do that in a separate video.